Welcome to another Macular and Me session. It's our second stream these days with the AMD on the third Tuesday, but we have a sort of a more working age focus, so it's about conditions that affect sort of younger people as a whole, and we talk about aspects of those things. So we're joined this evening by some lovely people from researchers and a genetic counsellor from Northern Ireland, who I'll introduce in a moment. As always, we're on a full webinar package, which means you can't unmute yourself or share any screen. So all you got to do is settle back, enjoy the conversation and the update from our speakers. But we really encourage you to join in. So if you want to ask the speakers any questions, just pop them in the chat, and they'll be read out a bit later on after the presentations. As you may or may not know, these webinars now get turned into podcasts, which is available on all good podcast packages or platforms rather. So you can listen back at your leisure if you don't have a chance to listen to it all this evening. And also, as always, this will also be on the website in a week or so after it's been sort of topped and tailed and tidied up with a bit of editing for the website. So we're joined as normal, really, as usual, by Jerry, the research manager from the Macular Society. Hello, Jerry.
Hi, everybody.
How are you doing? Are you okay?
Yes, fine, thank you.
Perfect, brilliant. Okay, well, maybe we can have a little chat about some Macular Society research bits and pieces if we have a bit of time at the end.
Yep.
And also, there's a patient who's my PA support worker. She's just in the background, sort of making sure everything runs smoothly. So this evening we've been joined by two people, which is unusual but great. Less time I have to talk. It's brilliant. Two of you from the Northern Ireland Research Institute. I hope I got that right. I read it about 4 times on the website, but I'm a bear of very little brain and I forget things really quickly. So we're joined by research optometrist Shelley Black and her colleague, a genetic counsellor, Claire Kirk. So good evening, guys. How are you?
Good evening. Hello.
Hello. Thanks for having us.
No problem. You're more than welcome. Well, I think without further ado, we've done the housekeeping. I'll just put myself on mute and I'll get over to you guys. So thanks very much.
Thank you. I'm just going to share my screen here. Let's see. So hopefully you should all be able to see my screen here.
Yep, looking good.
Yeah, all good. Excellent. So yes, like Colin said there, my name is Shelley Black. I'm one of the research optometrists at the Northern Ireland Clinical Research Network obviously in Northern Ireland, and I'm joined by the lovely Claire Kirk, who's our registered genetic counsellor. Tonight, we're going to be talking to you about inherited retinal degenerations, also known as inherited retinal diseases or dystrophies, depending what terminology you use, and also some of our macular research that we're doing. So I'm going to talk first, and then Claire's going to give you all the good stuff, and then I'll kind of wrap it up at the end, if that's okay. So let's see. Okay, so first of all, I'll just give you a bit of a run through of the order of how we're going to talk about things. So first little bit is just really why research is important, not only to people with vision loss, but just in general, really. And I think we've learned a lot about that over the last traumatic few years with COVID and all the vaccines, etc. Also, then I'm going to talk about the NICRN and what we actually do and what our roles are. And then Claire, I'm going to pass over to our lovely Claire, and she's going to talk a little bit more about the IRDs, the importance of genetic testing, and it'll be back to me again just to talk about some of our current studies and how you can get involved in research if that's something that you're interested in doing. So first of all, the reason why research is not only important but necessary is to learn more and to answer questions about the unknown. So it's not just about a new treatment and how it works and if there's any side effects, which is I think what maybe most of us think about when we think about research. It's also to maybe monitor a disease over time, because maybe there's not enough information to then develop a treatment. So that's really a key thing that we have to look at. These studies would generally last a bit longer. Maybe it could be 5 to 10 years, depending on the type of the disease and the type of the usual progression. As well as that, there may already be a treatment, but research is important to find out, is there a better treatment? Is there one that's more efficient? Is there one that's more cost effective? With the National Health Service, as it is at times, if anything can reduce patient waiting lists, reduce time and be more efficient, then sure, we're all winners with that. And then finally, when there isn't a treatment currently available for a disease, research is important to see if there's any kind of device or anything that can help improve quality of life and reduce symptoms. So the Northern Ireland Clinical Research Network. So it's basically a delivery platform for research. We work within the NHS, within the Belfast Health and Social Care Trust at the Royal Victoria Hospital in Belfast. And because we are in an NHS setting, it means that we can provide patients with the opportunity to participate and benefit from the latest clinical research. And the results from the studies that we perform can help advance clinical practice and provide an evidence base to improve treatments. So the NICRN doesn't work alone, and we partner with the universities within Northern Ireland. That would be Queen's and Ulster University, and then also in their wider partners in the UK with the UK Clinical Research Collaboration, Public Health Agency, and obviously patient and client councils as well. So the network doesn't just look at vision research, it covers all areas. So child health, critical care, diabetes, primary care, respiratory health, etc. There are many, many more. But this is the NICRN vision team. So not just me, if it was, not much we get done. We have in the bottom left-hand corner, we have the Professor Jonathan Jackson. He's our consultant optometrist. And on the right, we have Professor Julie Silvestri, who's our consultant ophthalmologist. They're both the leads for our vision team, and they're the ones driving the vision research within the Belfast Trust. Within the team, we have obviously optometrists like myself, we have research nurses, we have ophthalmologists, we have imagers, admin staff, and of course, you'll see in the right-hand corner, our registered genetic counsellor, Claire, who she kind of doesn't work in the NICRN, but she definitely gives a lot of input and helps with many of our studies, whether she wants to or not. Claire, isn't that right? So the areas that we cover, I think at the minute we've got maybe about 12 or 13 different studies currently running, where we are either actively recruiting patients or whether we're in the non-recruiting phase where we're treating patients. We don't just deal with macular eye disease, we also would do retinal vascular disease. So if there's any vein occlusions or anything like that, dry eye, glaucoma, refractive error, and obviously, as Claire is going to talk to us about next, it would be our inherited retinal degeneration. So I'm just going to pass over to Claire, and Claire, just you tell me when you want the next slide.
Thank you, Shelley. So Shelley has, I've put a little thing up here on the left-hand side of the screen, which some of you may be able to see. It's made up really of squares and circles. That's a family tree, and that's like my pen and paper of my work. In genetics, we take a family history from patients, and we do that for a number of reasons. Mainly, of course, to find out who is affected in the family with the inherited retinal degeneration. Are there some family members who've maybe been lost to follow-up and who we can get back into the system, the health system? We find out more about how the gene is inherited through the family tree. And, it also sets up rapport with patients, I find, because, once we start chatting about a family history, then, you get to hear the human side of things, the stories, about the previous generations and so on. So it's essential to our work, and it's the first port of call for talking to any patient, is to go through as well as their own clinical history, is to talk about the family history. Now, Shelley has put a photograph of an eyeball, I believe, beside that. Now, that's not my area, Shelley, but I think it's what you would call a quote unquote a normal eye, is it?
Yes, that's right. That's right. Normal retina. Yep.
That's a normal retina. So, very good. So, Shelley, if you could just get the next slide for me, please, that would be brilliant.
Thank you.
So, in terms of genetics, then, I've been doing the ophthalmology work for the inherited retinal conditions for about over 4 years now. And what has amazed me about ophthalmology is actually the number of genes involved. So, you know, for other genetic conditions like cystic fibrosis or Huntington's disease, there's only one gene. That's it. That's the gene. But for these retinal conditions, we know now that there are over 300 genes. So some of these genes are actually incredibly rare. So when a patient finds out what their family gene is, sometimes they'll say to me, oh, what do you know about it? What will I be like in 10 years or in 20 years? For a lot of cases, that is actually almost impossible to tell. There are some conditions where the gene is more common, like Stargardt disease. So the main gene there is ABCA4, and somebody in America has done a lot of work on the natural history of the ABCA4 gene, and they're actually able to pinpoint progression or let you know how your condition will progress with certain mutations. So for example, for some of the risk factors, they know that you won't be affected till middle age and your presentation will be quite mild. So there's so much to learn about the genes, but the more rare that the gene is, well, the more actually important that you are to us, to the community for research and so on, and the more you can help us. And again, that's where the family history comes in, because we can look at the presentation of a gene in the family history, not just in one individual. Colin mentioned earlier that this chat really is for people of, he mentioned working age, and indeed the inherited retinal conditions are the leading cause of vision loss in people of working age. So that's between 16 and 64. So there's not just the personal implications and the family and carer implications, but it's a huge loss actually to society as well in some ways. So these conditions are caused by what we call pathogenic or likely pathogenic variants in genes. And they're critical to the function of the retina. And as I mentioned earlier, there are currently over 300 genes that we know about. So Shelley, could you move on, please, for the next slide? So just to give you an idea overall, the inherited retinal conditions are rare. This is what we call a rare condition. We know that 1 in 3,200 people will have an inherited retinal dystrophy. The EU defines a condition as rare if it affects fewer than 1 in 2,000 people in the general population. Now, don't despair and think, oh, you know, they're going to forget about us because this is a rare condition. But as people say, rare conditions together are actually common. And this is why we've got the National Rare Disease Office in Dublin. There's one in Scotland. We'd love to have one in Belfast, and that will happen at some stage. There are more in England. So, but with inherited retinal conditions, we know more than 2 million people are affected worldwide. So, Shelley, please. So there may be quite a few of you who've never had genetic testing, and I know quite a few people get lost to follow up at the ophthalmology clinic, because for a lot of people, I suppose when you've been a few times, you maybe feel, well, they don't do very much. They take a few measurements and, you know, they're helpful if I have a sore eye or something, you know, along with my condition. But for a long time, I remember Professor Silvestri saying she used to feel quite bad. But genetic testing has come along and it's given us, it's really, really important actually. So if there's been no genetic testing in your family, we would recommend that you get referred for genetic testing through your ophthalmologist. Now, when we start testing in a family initially, we always start with somebody who's affected. And that person has what we call a gene panel test. So the laboratory will actually, it's like a needle in a haystack. They will look at over 300 genes in your sample. And if the familial gene is identified, we can then offer cascade testing or predictive testing to other family members. So if you find out what your gene is, we can then offer testing to your siblings, your children, and so on. Well, why would you do it? It confirms a diagnosis. It gives you a reason why this happened. It also refines the diagnosis, because even though we have this very, very broad terminology of inherited retinal degenerations, they are quite different in their own ways. And a gene actually makes it very refined. We can also establish the mode of inheritance, and that's important for other family members or for people who want to have a family and want to know the risk to their children. And leading on from that, then we can make referrals to the genetics service. There is potential there for something called pre-implantation genetic testing. So it's a form of IVF, and only the unaffected embryos, or the embryos that don't have the gene change, are implanted in the womb. Now, that's not for everybody, but again, it's down to your own personal history of your condition, or maybe what you've seen in family members in terms of their condition. But as I say, it's not for everyone. But it is important to know that it's available. But it's also essential to have your genetic report and your gene prior to joining clinical trials, and especially gene therapies. Thanks, Shelley. Thank you. So I'm just going to say something very brief about the chromosomes. We have 23 pairs. 1 to 22 of those pairs are called autosomes, and they're pretty much the same in men and women, chromosomes 1 to 21. And so when you hear a geneticist saying your condition is autosomal, it means that your gene is on chromosomes 1 or chromosomes 2 or 3 or 4. It's on a pair of the autosomes. Then different to that are the X-linked conditions. The 23rd set of chromosomes are the sex chromosomes. For females, that's XX. For males, that's XY. And for genes on the X chromosome, that condition is X-linked. Thanks, Shelley. So we've got a wee pictorial representation here of autosomal dominant inheritance. Now, for this condition, as I said, our genes come in pairs and our chromosomes come in pairs. So the genes are on the chromosomes like a little string, like a necklace. Each bead is a gene, and it's on that necklace, which is a chromosome. So we've got 23 pairs. One comes from dad in the sperm, one comes from mum in the egg. So for autosomal dominant, you just need one of your 2 chromosomes to have a little alteration or spelling mistake in order to be affected with the condition. So on this representation here, one of dad's 2 genes has an alteration and he is affected with the condition. So each pregnancy they have, whether it's male or female, will inherit a working copy of the gene from mum, and it's 50-50 whether they inherit their dad's working copy or the one with the alteration. For an autosomal dominant condition, we're able to tell people that for each pregnancy, there's a 50% chance that it will inherit the gene change and will be affected. Thank you, Shelley. Oh, Shelley has put up some photos here. So these photographs, apparently they represent the RHO gene, which is dominant, causes retinitis pigmentosa. So Shelley, you might be able to say the one on the left there has the signs of birth.
Yeah, so these are both the right eye of 2 different patients. The one on, as you look at your screen on your right hand side, is a healthy eye. So you'll see it's a nice orangey red color. The nerve, which is the little like yellowy dot, it's a nice bright color on the healthy side. If we go over to the left, there are these very these little black speckles that are surrounding the middle part of the eye. These speckles are called bone spickle dark deposits, and they are very characteristic of patients with retinitis pigmentosa, and particularly this RHO gene. The nerve, instead of it being this more warm kind of color, it's a bit paler, and and I'm sure anybody in this call who has the condition, they'll know that the difficulties they generally have would be seeing in dimmer light and at night initially, maybe with some blind spots in the peripheral vision, and later on will notice some issues with their central vision as the central vision starts to degenerate a little bit as well. Generally, the symptoms would begin in adolescence and young adulthood, and then progresses then over time a little bit with that. So yeah, that's that. Claire, I'll pass you back.
Thank you. That's really helpful, because I've seen bony spicules written in clinic letters and things. I've never been quite sure what that meant. So that's very helpful. So we're sticking to autosomal inheritance, so the autosomes. This time, I want to tell you about autosomal recessive inheritance. So if you remember with the dominant type, you just need one of your two genes to have a little alteration to be affected. With the recessive type, both of your genes need to have an alteration. We used to use the phrase mutation, but it's sort of more nuanced now. So both copies need to be affected for you to be affected with the condition. And I'll go back to one of the most common genes, ABCA4. That's how this condition is affected. So if you have that condition, you can bet that both of your parents are carriers, what we call unaffected carriers. So with the autosomal recessive conditions, these conditions just seem to come out of the blue. So people will say, but, you know, when they have an affected child, you know, we don't have a family history of this condition. We've never heard of it. Why has it happened? And it's happened just because one unaffected carrier has met another unaffected carrier. They didn't know they were carriers, but every time they conceived a pregnancy, there was a 25% chance that that pregnancy would inherit the altered copy of the gene from dad and the altered copy of the gene from mum. And that's simply it. So that little group of children or siblings will be the only section of the family with affected family members. You will have carriers on both sides of the family who won't know they're carriers, but it's when you get one carrier having children with another, you start to see the condition. Now, some of those children will also just be carriers, so they'll be unaffected, and some of them won't even inherit an altered gene at all. The one thing, the ensuring thing, I suppose, with knowing about the inheritance pattern is if I meet somebody who has Stargardt's and they know it's the ABCA4 gene, I can let them know that, look, your children will be carriers because they'll have to inherit one of your altered copies, but they'll get a wild type normal copy from the other parent. ABCA4 will be the most frequent gene in the population out of all these inherited retinal conditions, but it's not frequent enough to warrant carrier testing in the general population. So we don't actually do that. We don't offer carrier testing. It's really not needed. It would just be an issue if you knew that somebody, you know, was going to marry, say, a first cousin or something like that. And that doesn't happen a lot, but I have come across it in clinic. And when that happens, then we do carrier testing for the cousin or so, and so on. So it's just a bit different from the dominant inheritance. So thank you, thanks. And again, here we've got photographs. This is for the ABCA4 Stargardt. Shelley.
Yeah. So in this slide, both photos are of the CMI, but photo B on the right is just a more magnified version of it. So if you can, so there's little dull yellow deposits just surrounding a paler area, just in the middle of the photograph, just around the macula. These deposits are called flecks and are a hallmark of Stargardt's. And, you know, again, anyone with Stargardt's will know that typically it's a central vision loss, which tends to worsen over time as larger parts of the macula becomes damaged. However, the severity of the visual loss and the rate of decline are highly variable with this condition. So, so yeah.
Thanks, Shelley. Yes, that's right. Shelley said the clinical presentation is highly variable, and that comes down to the type of gene change that you carry and the combination of gene changes in both of your genes. So it is quite fascinating. And There's also a new science that, if you like, it's called eye to gene. So it's quite amazing that an ophthalmologist can look at a photograph of an eye or an image and can actually say, well, that's Stargardt, so that's going to be such and such a gene. You know, so when I started, I found that really fascinating, that the clinicians can really make a very good guess at the gene the family might have just from clinical pictures. So this is the last form of inheritance here. So this is, I mentioned before about the sex chromosomes, the X and the Y. And we talk always about the X chromosome because the Y, which the males carry, has actually got very, very few genes on it. So it's all about the X chromosome. Now that's a very busy picture there on the left, and I don't like it. So I'm just going to say that when a male has an X-linked condition, his one and only gene, because remember, he doesn't have a pair, he only has one gene. When that gene has the alteration and he goes on to have children, he cannot pass it on to any sons that he has. So that is because he only passes on the Y chromosome. So again, that's the importance of knowing where the gene is and how it's inherited. We can reassure a male carrier that he won't pass it on to any sons. Now, on the flip side, his daughters will be carriers, but with many X-linked conditions, we find that the women are not as severely affected, and they're affected much later in life. And we think that's because the second copy of the gene on their second X chromosome compensates for any loss, you know, through this altered gene. So the girls will be carriers. Now, in the second part of this wee diagram, we can see mum is actually the carrier here. In this case, it's more like autosomal dominant inheritance. Boys and girls will have a 50-50 chance of inheriting the X-linked gene from her. So I suppose the other thing to say about X-linked inheritance, the main gene that we work with is called RPGR. And this is sort of flipped it around for us. We're finding the female carriers are also quite severely affected, and at a young age as well. So as I found over the years with genetics, there are no hard and fast rules. There are exceptions to the rules. And so we know that RPGR doesn't behave in this way. And we think it's just that in the cell, the working copy of the X gene is accidentally switched off. So you have expression of the altered, or the normal copy switched off. So you have expression of the altered copy, and that's not the way it's supposed to be. So we think also there's a lot of background genetics. So we focus mainly on monogenetics, or one gene, a big gene in the family. But there's something now coming along called personalized medicine, where there's also a genetic background. There are other genes that we can't disregard. We don't know enough about them yet. And that's why within families, you see such a lot of clinical variability with some of these conditions. The genes in the background are playing as well. And so eventually, as well as looking for the main gene in your family, we may get to a situation where we can look at the background genetics as well and see how that plays in to the environment in the cell and your retina and so on, your condition. So, sorry, thank you, Shelley. Shelley's put up a few more photos now of an X-linked condition in the eye.
Yeah, so these slides are just showing choroidemia, which is in this case caused by the CHMG. So again, on the right hand side we have healthy retina, and then on the left hand side we have retina affected by choroidemia. So anything that's kind of pale in the retina generally means that it's not really working anymore, that it's wasted away. So the only part of this eye which is healthy or seeing well is right in the middle in the macula. So people with this condition will very much have tunnel vision, and how this varies again, the severity of the symptoms can vary between patients, even within the same family, with this particular one. So yeah, I'll move on to the next one, Claire.
Thank you. Yeah, so I just put this up. It's a bit geeky, I suppose, a bit nerdy. But I suppose what I'm trying to say with this slide is, look, the family tree really helps us to consider a mode of inheritance for the family, but you do not really know what's going on until you actually do the genetic test. So just looking at that family tree, you would say, well, this is a sporadic case. What's the inheritance here? Well, believe it or not, it could actually be dominant, even though I talked about how dominant genes are inherited. But what's actually happened in this case is this patient here who's affected, that genetic alteration actually occurred spontaneously at conception. So there's no family history, but it has started for the first time in this person. And the importance about knowing about this de novo genetic alteration is that going forward for this person, any children they have will be at 50% risk of inheriting that alteration. So there's the importance of doing a gene panel for that person and working out how this genetic alteration came about. So it's de novo in this person. It wasn't inherited. It happened at conception, but going forward, all of that person, his children, will have a chance of inheriting, 50% chance. So the other thing is it could be autosomal recessive. Both parents are unaffected carriers, or it could be X-linked. So each of the females, his mother, his maternal grandmother, and so forth, were X-linked carriers who didn't develop the condition, but have maybe when they are examined, have very, very subtle signs. So in boys, though, unfortunately, it tends to be quite severe and quite affected at a young age. So anyway, thanks, Shelley. So I don't have too much more to say on this. You'll probably be quite glad, but I just wanted to give you, I think it's very important when we're counselling people who are having gene panels. It's important to let them know that we may not find your gene. And that's down to technology, really. There are parts of the gene which are not very well characterized and are quite hard to access. So, a lot of people will be hoping for a positive result and find out what their gene is. But I just did a wee snapshot of our results for 145 patients, and it was over a year, 2019 to 20. And out of the people who had gene panels, 79 of them got a genetic diagnosis. So that's 54%. 55 of them got a negative report. So the causative gene was not identified, but that doesn't mean that your condition is not genetic, so you're at a bit of an impasse there until the technology improves and we can look at other parts of the genes. But there's a very small percentage, 8% of people will have what they call a variant of uncertain significance. And that's a kick in the teeth for the professionals involved as well, because what's happened is the lab has identified a good gene and there is an alteration in it. But there isn't enough scientific evidence to say whether it's caused the condition or it's just one of those benign changes that occur in all of us across the general population. There's just not enough evidence. And part of that is because these genes are so rare and they maybe haven't seen that particular alteration before. So these variants of uncertain significance tend to be reviewed every 2 or 3 years. So it can happen. If you do family studies and try to follow this change through the family, through affected family members, you can sometimes upgrade that classification to likely pathogenic. So it's just to manage people's expectations there. Thanks, Shelley. And this again, it's just, these patients where we did get a diagnosis in 79, it was in 19 different genes, the most common of which, I've mentioned them before, ABCA4, we had 34 patients with a diagnosis. The USH2A gene, which is also autosomal recessive, so carriers aren't affected. It causes Usher syndrome type 2. 12 of our patients had that. And then as you go down, you can see how rare some of these genes are. Like quite a few of them only happened in one patient. So the next one there is RPGR, that's the X-linked one. We have 6 patients with that. So it's quite a range. So thank you, Shelley. And without stepping on Shelley's toes, I did think I'd just put this in about gene therapy because I know these things seem to move very, very slowly. But there is actually a genetic, a retinal condition where there is gene therapy. So there's a condition called Leber congenital amaurosis, and it affects really toddlers. You know, by the age of 18 months to 2 years, they're almost blind. But for that condition that's caused by this one particular gene, RPE65, there is a gene therapy, and it's funded by the NHS. Now, we've been trying for years, as Shelley knows, to find a patient in Northern Ireland with it, and we haven't. We think there are 3 patients in Dublin. So it's quite an amazing thing, but it does give us a great deal of hope for other genes going forward. Thanks, Shelley. And just finally, there's a very smart registrar in working with us in Belfast. He did some work over in Murfields in the eye hospital, and there's a lovely app he has. You can download it on your phone. It's called Gene Vision. And it goes through all the different genes. And actually, Shelley had a look at it when she was preparing her slides, and that's where she picked up the photographs, you know, for the different for the eyes, for the different genes. But it's a lovely app. It's been developed for patients. It's got information about support groups and so on. And I really recommend that you have a look at it. So that's it from me. I think so.
Yeah, back to me. So yes, just as Claire was saying, that website it's really, really good, particularly if you've had genetic testing and you know what your condition is. Even if you don't and you know the name of your condition, you can click on the condition on that website and it will have the most up-to-date information if there are any current clinical trials going on. So you can kind of link with that and see if they're recruiting patients or if there's any way you can participate. So like I said at the start, Claire's done all the heavy work and I'm just going to wrap things up here in the last few slides. And I just really want to talk about briefly a few of the studies that are currently going on in the NICRN at the minute. I just wanted to focus on ones that are more macular based or inherited retinal degeneration based, and just to show you a sample of the different types. So the GALTOS study, this study is looking at people with a more advanced form of macular degeneration, so geographic atrophy. And atrophy is from the Greek word meaning wasting away. So it's where part of the retina kind of starts to not work at all anymore. And that's what gives us that kind of that blurred vision. So currently in the UK, there's no approved therapies to treat this. Now, there have been some that have been approved in the US, but that hasn't reached us here. So this is an example of what I was talking about earlier, where sometimes we don't have enough information about a disease or why it affects some people more than others. So with this study, what we are doing is just monitoring the disease over a period of time. The patient doesn't even have to come to see us in the research department. We're just we're observing their general care and their general visits, and it's just to see, why, once they get all the information from all the different sites, because it's not just us, it's many sites across the UK and Europe that are doing the study, then they will hopefully try and find out why one group is different than the other, and hopefully that will lead to further research. This next study is called the Tiger study, and you will notice that studies all have these very catchy names, and that's on purpose, because catchy names, that people remember them compared to the very technical name, which I'll not go through, but there's about 20 words to it. So the Tiger study, I just wanted to give an example of a macular condition that's more an acute one, so it happens more suddenly. In this study, the patients that are affected have a submacular bleed. From the photos, you can see there's a really heavy blood clot just centrally. And the OCT image on the right, that's just showing that big kind of hill, which is just where the clot of blood has kind of gone underneath the macula. It should normally be a valley as opposed to a hill in that picture. This study is comparing two types of, it's comparing a new treatment versus the standard of care. So this is an example of trying to find a more efficient, better way to treat a certain condition. So in this case, it's it's comparing the standard eye injection to the eye to help the little blood vessels to stop bleeding versus the injection plus surgery, where a clot busting drug is inserted at the same time along with the gas bubble to take the blood away more quickly. I have a video on the next slide, and if you are squeamish and don't want to see horrible things happening to you, I would advise you not to look at this next bit. But if you're interested to see what an eye injection looks like, just to be more reassured that it's not as scary as what you think, then just for the next minute, I'll let you watch. So bear with me. There is sound in the video, but you'll not be able to hear it. So I'll just kind of talk a little bit about it. So hopefully you can hear me still. So basically, this lady is one of our research patients, and she is getting one of the eye injections. The surgeon has just marked on the eye where he's going to do the injection and asked the patient to look up and to the left. And he has just popped the injection in. So that's 1, 2, 3, 4, 5, 6 seconds it took to get that done. All done. And he asks the patient how she feels about it. And if you can see her hand, and she basically just says, it's not painful, it's just pressure. So I was trying to show my husband this image during the week, and he almost vomited. So there we go. I'm not even joking. So last study I'm going to talk about is obviously the MAGVIEW study, which we're very thankful that the Macular Society has funded. In this study, it's an example of where there currently isn't maybe a treatment for a macular condition. So in this study, it's not just people with macular degeneration, it also includes people with inherited retinal diseases. And the study is aiming to see if intraocular magnifier can help patients' reading ability. So if somebody's already had their cataract removed, they'll have an artificial lens in their eye already, and the implant just slides almost like a piggyback system on top of that lens. The central part of the lens provides about 2 times magnification, and then the outside of it is clear. So the point of that is so that it still gives a patient distance vision while also helping with the reading, because as we read, our pupils naturally shrink and should be focusing onto the more magnified part of the lens. Alongside this study, we're also testing out a contact lens on a similar principle with the magnifying area in the center and clear bit on the outside. And with the contact lens, we're trying to see whether it's comparable to the lens implant, so that if people don't want to have surgery, then that's another option for them. Now, because it's obviously, if some people might be listening going 2 times magnification, sure, that's not very much, and you're right, it's not loads. So the patients in this study generally would have a more mild to moderate visual loss, rather than we wouldn't be recruiting people with more severe types of vision loss for this study. So last slide, really. So how can you get involved in research? So the Macular Society, I've been looking at their website, are very good in updating you of what's currently available, what's going about, and obviously are funding research all over the UK, which is absolutely fantastic. Without the funding, research can't happen and we can't move forward. But if you are, obviously everyone on this call isn't from Northern Ireland, so if you're living in the UK, the people that do the research are clinical research networks like what we do in Belfast. They're all across Scotland, England and Wales. You can contact your local research network, your research facility, your clinical trial unit, any of your universities, and just ask if they have any studies running with regards to your condition. Now, just an important point, the thing with clinical trials or with any study, they do generally have quite a strict what's called eligibility criteria. So even though you may have the condition, you may not fall in line with what they want. It may be you have to have a certain amount of vision loss or impairment, like I mentioned in the MAGVIEW study. You may have to only have an eye condition that's stable, that you can't be receiving injections for, or you may not be able to participate in the study if you have other conditions like diabetes or hypertension, etc. But don't let that dishearten you. There's always something coming up, and even just letting the researchers know that you're interested means that if anything does come up, they will be the first to contact you. And just following on from that, if you are in Northern Ireland and you are not known to us and you are interested in taking part in any research, please drop us an email at the Vision Research Team at belfasttrust.hscni.net, or give us a call on the number below. But thank you for your time tonight. I know we maybe went on a little bit longer than what we expected, so apologies for that. Thank you for listening.
No, it's been very interesting. I've really enjoyed it. Thank you ever so much. It's it's brilliant. I think understanding genetics and sort of what you guys are doing over there as well over in Northern Ireland is brilliant. So thank you for your time. I think we had one question or two. I heard a couple of mumblings come through on my screen reader. So Jerry, did we get any questions?
Yes, we've got a couple, both from the same person, but they're not particularly related. So the first question is, it's to Claire. When you talk about people needing to know their genetic mutation alteration in order to take part in gene therapy or go in a clinical trial, can you explain why?
It's a bit like matching pieces of a jigsaw puzzle. The gene therapies are targeted very, very much towards that specific alteration or spelling mistake in your gene, or specifically towards that gene. So it's like a lock and key. If you imagine it's like a lock and key. So the idea is either to remove the genetic alteration or to flood the retina with working copies of the gene. You know, so if you don't know what that gene is, then you can't match the jigsaw puzzle. You can't put the lock and the key and match the things together. I don't know if I've made that. The gene therapy is specific for that gene. No.
So I think probably it doesn't stop anybody taking part in any studies. It's just if it's particularly gene therapy where it's targeting the actual gene, but they could still take part in a study where they're looking for people with ABCA4 gene. It's just the actual therapy where they need to target the gene. Then that's where you need to know what gene it is you have, and
I've seen the eligibility criteria for clinical trials, and there was the Stargardt one not very long ago where you couldn't find out what your alteration was through the trial. You had to know it already for them to consider you for the trial. So you had to have had that genetic diagnosis before you would even be considered. You couldn't just say I've got Stargardt's and they would look at you.
One of the reasons is I've mentioned ABCA4 quite a lot, but there is another gene which causes Stargardt's disease, PRPH2. It's inherited in an autosomal dominant fashion. So if a clinical trial, or particularly a gene therapy, but if it is targeted towards a particular gene, it wouldn't be helpful for somebody with Stargardt's, which was caused by the other gene. Is that, you know, I don't know, Shelley, if you want to comment
on that. No, I think like that's a big reason why there's a lot more funding and stuff going into people who haven't got genetic testing to to find out. Genetic testing, it does take a long time to find that gene, and if a study has already been given the go-ahead, they might have a limited time frame in which to see the patients. So if it's going to take maybe, I don't know, Claire, can it be up to 6 months, 4 months? I don't know. It can be quite a long time for some.
You know, basically, if you've got your genetic report and you know your genetic alterations, you're good to go. You don't have to go through that genetic protest or the genetic testing initially. We've been very fortunate in Northern Ireland. We have had funding for gene panels, and we've been able to get it done through a commercial company. Now that's funding is finishing in 6 weeks, but I mean their turnaround time was just phenomenal. It was between 6 weeks and 8 weeks. But if you're having a gene panel through the NHS, it's a minimum of 9 months, but generally it's about a year to get a result. So this is why it's really important, I think, to have all that background work done, and then if something does come up, like something that targets ABCA4, as I say, you're good to go. If, as Shelley said, you'll fulfill all the other eligibility criteria, maybe your vision has to be at a certain level and so on. But yeah, that's the importance of having your genetic report.
Lovely, thank you. So another one's come in. I think this one probably for Shelley. Somebody's been diagnosed with bilateral macular drusen and some drusen nasal to the optic disc. Now, drusen is something I know of in relation to age-related macular degeneration. So can you recognise that as a diagnosis related to an inherited disorder?
So what is it they're asking? Is it what is it? Or
I would like to know more about this bilateral macular drusen.
And it's bilateral macular drusen, but it's also at the disc as well.
A few drusen to the optic disc, and they've just popped in the chat that they're 30 years old.
Oh, okay.
That's very young to have drusen.
Yeah, it's quite young to have drusen, but it's not completely unheard of. We have seen people in the past. I also work in community practice as well. And there would be maybe the odd person you would see who would have more like a juvenile kind of drusen, but their vision isn't necessarily, severely affected. That's just the way they're made up. So it wouldn't necessarily maybe come under the inherited retinal dystrophy kind of umbrella. I don't know, because I don't know the person, I haven't seen their images, I can't really say lots about it. It wouldn't be as common. It depends if the drusen is near the disc, then again, that would be that kind of would fall in line with any drusen that's more associated with when you're younger, because it wouldn't necessarily just be at the macula, it could be a bit more scattered. If it's optic disc drusen as well as that would be, I don't know if I've come across that. That would be maybe a bit more. So again, I can't really comment on without seeing the images or seeing it exactly. But yeah, it's all kind of very dependent on the vision. If the person is unsure and wants to know more about it, go into, don't be afraid to ask for more information. I think sometimes we get into the clinic room and we're told something and they go, is that okay? Here's a leaflet. And you're like, okay. And then you walk out and you go, I don't know anything about this. I don't know, whoever is giving you that diagnosis, don't hesitate to go back and just be like, could you please give me a little bit more information about this, or direct me to some support where I can find some more information about that. I think
Absolutely.
that would
be one of the things that we talk about all the time is, if you've got those questions, write them down, and the next time you go to the eye clinic or whomever, just take those questions with you. So you don't forget to ask the relevant questions that you think of over dinner 3 weeks later.
Yeah, because it's the worst thing. And like us as professionals, that's what we're there for. We're not there to, yes, there's busy clinics, whatever, but you're still worth the time, and never feel like you can't ask the question, because you always can, and you deserve to know an answer. And if they don't know an answer, they'll know somebody who can. So yeah, I would definitely say ask the question. And even if it's after, don't wait till the next appointment, ring up, ask. Be that person.
Be that person.
Yeah.
It's your condition. You need to know all about it.
Yeah.
Understanding is really important. That's why diagnosis is so important to people. Okay, next one is for Claire. So this person has had genetic testing, and their gene where the fault has been identified is GUCY2D, and it's autosomal dominant. Do you know anything about this gene, and is there any research being done?
Well, the first thing I can tell you is that it is rare, and we do have a family in Northern Ireland, but unfortunately, when we did genetic testing, the result came back as a variant of uncertain significance. And in that situation, we tried to contact other labs, research groups, to try and find out if they know anything else about the variant. We've not been able to find anybody else with that exact variant. There was one patient, I think, with a slightly different variant, who had a slightly different clinical presentation as well. So I'm so sorry in terms of how it might affect you or other affected family members. I wouldn't be too sure. I know with our family, it is autosomal dominant in the family quite clearly, and everybody who carries it is affected. But I can try and find out a bit more for you if that helps.
I just had a quick look up on the gene Vision
In
website
Vision.
there, just for a quick scan. And it just says, so it has a wee topic of therapies under research. And I think there's some phase 1 and 2 trials maybe going on with that at the minute. But if they have a wee look at that website and there's a link for the studies, then that would maybe give a little bit more information about the current therapies or currently trials that are under research at the minute, if that's helpful.
Thanks. Thank you, Shelley.
I'm no expert in this area, but I can see that the research is going away from specific gene treatments, gene-related treatments or mutation-related treatments, to a more a broader form of gene therapy, or broader form of treatment that will be able to be used for a range of mutations. So because if we were trying to develop a therapy for each specific mutation, so many of them that would take forever. Thinking more broadly to try and find a treatment that would impact a wider range of conditions.
Yeah, that would be ideal.
Yeah.
Well, I'm very aware of time. We have managed to get through our hour quite rapidly, which is always good. Just leaves me a chance to say thank you, Jerry, for coming along as always and doing the questioning, answering the questions. It's brilliant. So thank you very much. But a special thanks to Claire and Shelley for taking time out of their evening to come and chat with us this evening. So thanks, guys. It's been really great, really informative, really interesting. So
thank you.
Thank you for having us.
Thanks. No, absolutely no problem.
I just want to say that
question.
I've just popped a link into the chat for Be Part of Research, which is an NHS website which lists all the clinical trials underway in the UK. And
brilliant.
So that's updated on a regular basis. So
where have you put that, Geraldine? Where is it?
In the chat.
Okay. I think there's another question in the chat. There might be another question.
I've gone and put it in the only to hosts and panelists. I apologise. I will redo that to everyone.
Be part of research,
There you go.
Brilliant.
To everyone. Cool. You can all see it. Sorry about that.
Excellent. All right. Well, in that case, we have drawn to a close. Only to say that the next session is on areas of Stargardt's disease. So back to the ABCA4 gene again, and we're beginning looking at light sensitivity, so photophobia, and on all those lovely swirly things that people get in their eyes if they've got Stargardt disease, photocia, or I think I've pronounced that correctly. So that's on November the 30th, and that's the last one of the year. So you have no excuses but to come along and learn some stuff. So it'll be really great. So again, thanks Claire, thanks Shelley, thanks Jerry, and thank you to patients for keeping an eye on everything in the background. So thanks very much and good evening.